Hello and welcome to the EASD 2020 Meet the Experts session on the introduction of a 7-day infusion set, Medtronic Extended Infusion Set and Reservoir. My name is Ohad Cohen. I'm a professor of endocrinology and also serve as the Director of Medical Affairs for Medtronic Diabetes in Europe, Middle East, and Africa. And with me to discuss the scientific background of this new infusion set is Dr. Sarnath Chattaraj, who is our Senior Director of Research in charge of the innovation in the consumable infusion sets. We will discuss the need of longer infusion set duration. We'll discuss the science behind the new set. And at the end, we will discuss the clinical evidence of these new sets that have been performed in trials. So what is the need for longer infusion set duration? Well, to understand this, we have to understand the path that insulin does from when it's manufactured in the glass vials till it gets to the subcutaneous tissue and absorbed to do its action. Well, it goes from the vials, it goes to the plastic reservoir. From the plastic reservoir, it's put into the pump. In the pump, it's pushed and squeezed by the plunger into the infusion set, and then it goes to the subcutaneous tissue. All this via Dolorosa of the insulin makes the insulin unstable, and at the end of the day, it causes the need for the patients to change the infusion sets every 2 to 3 days. And these issues with the insulin and the need to change the sets every 2 to 3 days made the critics of the pump and insulin delivery devices call the infusion sets the Achilles heel of the therapy. And as explained by Lutz Heinemann almost 15 years ago, that the need to be put more emphasis in significantly reducing the frequency of insulin infusion problems to increase the acceptance of the insulin pump therapy. That is, putting the patient needs in the middle of providing novel technology solution to the infusion set. And in order to address these issues, we have to take into account several topics on the infusion set. Number one, with the new innovations in insulin, automated insulin delivery system, more and more patients will use insulin pumps for their therapy of type 1 diabetes, increasing the issues with dermatological complications that needs to be addressed for them to have a good outcomes with the new devices. Infusion set that needs to change every 2 to 3 days cause dermatological issues that we need to decrease. Patients want to change less frequency, and this is obvious, less frequent changes a week. Now, if you go to a longer infusion set time, you increase the issues with the stability of insulin that needs to be addressed. Further on, by changing sets every 2 to 3 days, there's a lot of waste of materials and insulin that is in the infusion set that we throw away. And last, looking forward, having a combination of both a infusion set for 7 days and a sensor for 7 days will really improve patients' experience and patients' satisfaction. So it's not only that the critics were asking for innovation, it was also the regulator. And this is the FDA, more than 20 years ago, already brought up this issue in the discussion of insulin with the insulin companies, requesting to address the instability of insulin in pump therapy and urging to find solution to increase the survival of the infusion sets and the insulin in the infusion sets. Now, in order to improve survival or improve the longevity of the infusion set, we have to, first of all, define what is a set failure. Now, this was not well known in the past, and one of the originators of the definition of a set failure was Dr. Bruce Beckenham from Stanford, California. And he brought up the concept that a set failure will be defined as when you have unexplained hyperglycemia, let's say more than 300 milligrams per deciliter. Unexpected, that means more than 3 hours after a meal or when there was nothing specific that caused the hyperglycemia, and it's not corrected by correction bolus by reducing by more than 50 milligrams per deciliter, or that in addition to high blood glucose, you also have evidence of ketones. Lastly, if you have skin reactions that preclude you from using the site more. So those are the definitions of unexplained hyperglycemia that we want to prevent with novel insulin delivery systems. In order to address these issues and to solve them, I would like to have Dr. Sarnath Chattaraj explain to us what was the technology behind extending the wear of the new infusion set. Dr. Sarnath.
Thank you, Dr. Cohen, for the great introduction to why we need an extended wear infusion set. Hello to all, and thanks for listening to this session. My name is Sarnath Chattaraj, and I am an employee at Medtronic. Welcome again to the virtual EASD 2020 session. There has been a progressive increase in the number of people with diabetes in using continuous subcutaneous insulin infusion, also called pump therapy, since its introduction over 30 years ago. One of the challenges of pump use include infusion set failure, which can lead to hyperglycemia and diabetic ketoacidosis. Patients undergoing CSII as a means of insulin delivery are currently advised to replace their infusion sets every 2 to 3 days. There have been several research to understand the wear duration and develop a set that would last beyond this duration and reduce user burden. At Medtronic, we have also engaged in the research into longevity of the infusion sets. Now, let's start by examining the base of the research again. I think Ohad shared with you, and now let me go back to it again. Infusion sets are required for subcutaneous administration of insulin by a pump. The insulin is transferred from an insulin vial to the insulin reservoir, then connecting the insulin reservoir to an infusion set and delivering the insulin using an external pump into the subcutaneous tissue. To date, the infusion sets, which has either a steel or a soft cannula, are used for 2 to 3 days. It is thought to be that there are multiple reasons limiting the wear duration of infusion sets to 2 to 3 days. One of the reasons, as per insulin labeling from major insulin manufacturers, it is the insulin stability, the loss of preservative, and the occlusions are thought to play roles in shortening infusion set wait time. Like other researchers, we started our research in analyzing clinical studies to date as to what are the reasons that people with diabetes cannot wear the infusion set longer than 2 to 3 days. One of the leading research was published by a team from Stanford University in the USA, led by renowned endocrinologist Dr. Bruce Buckingham. His studies has been very comparable to the studies by other researchers, including us at Medtronic. Let's examine the numbers in context of this publication. The majority was assigned to unexplained hyperglycemia, which Ohad had already explained in his previous deck. Other reasons that contributed to the infusion set failure is removal due to pain, followed by skin infections, and failure to adhere to skin for longer duration. And lastly, but not the least, the failure due to accidental pulls. The challenges in the day-to-day life of a diabetic has included incidences where a cat at home chewing up the infusion tubing. We focused our research on primarily 4 elements: unexplained hyperglycemia, removal due to pain, skin infections, and adhesion. To assess the impact of the above on the wear duration, we at Medtronic developed a test model comprising a set of test methodologies for evaluating infusion set failure and assessing the causes of sight loss during CSII therapy. For the purpose of the assessment, we have used the 2 primary insulin delivered using Medtronic pump: insulin aspart, as in Novolog, and insulin Lispro, as in Humalog. 4 test methodologies were used in succession for the composite test model for this assessment. First, in an in vitro setup, the insulins were pumped and collected in glass scintillation vials. The in vitro study simulated as expected in closed loop therapy. These pumped samples, along with glass vial controls, were analyzed for chemical stability of insulin and preservatives. The results from insulin testing met pre-established specifications. However, a drop in preservative level was observed and was more pronounced as the number of days of infusion using the same infusion set increased. In the second test, the samples from first test, along with insulin formulations having lower preservative content than the label claim, were then analyzed for physical stability, primarily by dynamic light scattering. The insulin aggregates were found to have increased in pump samples and was correlating to lower preservative content. In the third test, these samples were then characterized for inflammatory responses. In murine macrophage cell, the ingredients likely presented during sub-Q insulin infusion, including fibrinogen, macrophage cells, and insulin, were tested in the cell culture study. There was no obvious cytotoxic response observed in any of the combination. The macrophage cell growth density and morphology looked normal. However, fibrous materials were observed in the cell culture with elevated insulin aggregates. Significantly more pro-inflammatory cytokines, the chemokines MIP1 alpha and MCP1, were detected in the supernatant media of the culture with insulin aggregates. The results indicated that elevated insulin aggregates resulted in increased inflammatory response from the macrophage cell. In the final test, we took these insulin formulations formulated with lower preservative content than as per label claim from insulin manufacturers, and then tested along with nominal controls in an animal model. The infusion set wait time decreased significantly and occlusions increased with lower preservative content. The results confirmed that insulin stability and preservative loss as one of the root cause behind infusion set failure and reduced infusion set wait time. In summary, these findings using the composite test model indicate that macrophage number, the inflammatory response, and device wait time were significantly impacted by loss of preservative and trace aggregates particles, among other reasons. So what we did at Medtronic, we employed this test methodology along with other tests to develop a novel technology for extending the wear duration of infusion sets. Our first mitigation was how to manage the failure attributed to insulin stability. So if you remember our infusion set, it has a cap that connects to the pump and the reservoir. So we developed and inserted a set of novel components in the fluid path of the infusion set. They are located inside the Medtronic proprietary connector that I just showed you that enables connecting the infusion set to the reservoir and the pump. It has been a privilege for us at Medtronic to collaborate with Dr. Bruce Buckingham for this research, who did a clinical study using the novel connector and he presented the data at this year's American Diabetes Association scientific session, which confirmed that we were able to mitigate a major part of the hyperglycemia using that connector. What we did then is actually move to the second phase of the development, and we made advancements in the tubing material to improve preservative retention. In collaboration with Convatec Uno Medical, a longer wear adhesive patch was also developed and was included in the design. User studies were performed for adhesive patch selection. I'm going to share that data in my next slide. The innovative infusion also features a pre-loaded inserter developed by Convatec Uno Medical that can help insert the cannula using one hand, thus helping patients place the infusion set in hard to reach areas. The infusion set, called the Medtronic Extended Infusion Set, along with a compatible Medtronic 7-day reservoir and Medtronic pump, was used in a clinical study. Ohad is going to share the data from that study later in this session. Let me briefly outline the adhesive studies. Adhesive performance on the body is highly complicated, as you all know. Although there are many standard tests available, no in vitro in vivo correlation has been established. The objective of the study was to evaluate adhesive performance and patient comfort of 3 new prototype adhesive patches intended for extended wear infusion set, comparing to the current on-market infusion set adhesive patches. 75 adults participated in this study, with each one blindly won 2 randomly assigned infusion set with patches from total of 7 variants. The primary outcome was determining survival rate at various wear time, along with secondary outcomes wear comfort and removal discomfort. We used Kaplan-Meier method to compare the survival rate versus wear time among adhesive patches. In this simulated use conditions, 2 new adhesive patches, variant 5 and variant 7, made the primary survival rate and the secondary user experience outcomes for use with an extended wear infusion set. Adhesive patch variant 7, which had 100% survival rate for greater than 7 days, was selected for use in the new Medtronic extended infusion set. So to summarize, what we did actually, we looked at the continuous subcutaneous insulin infusion and we categorized them into 2 categories. One was initial inflammation from insertion of the foreign body which came from the needle and the cannula insertions. And the second was sustained inflammation from insulin infusion. We actually stayed focused on the second set of the sustained insulin infusion. Needle and cannula can contribute to the inflammation as the cannula enters the subcutaneous layer of the body. Some research indicates that preservative, which possibly contributes to cell death, is the reason behind the side loss. The technology involved at Medtronic extended infusion set focused on fluid stability and dynamics that included insulin stability, insulin aggregates, and preservative loss. This was the hypothesis that was tested in the clinical study that Ohad is going to present. Thank you, and over to you, Ohad.
So thank you, Dr. Sarnath, for providing us the evidence and the scientific background. Now let's look at the clinical evidence. Remembering that fibrils formed in the infusion set and in the pumps are cytotoxic to the subcutaneous tissue and the tissue surrounding the infusion set. And we want to see whether the new infusion set can prevent this reaction and prolong the activity and the viability of the new infusion set. For this, the following clinical study was performed on the full set as been described by Dr. Sarnath. This was a single center prospective open label study with 26 participants for 1 month. All in all, we had 82 insertions with 78 wears to analyze. The results of the study was compared to the historical published data on the QuickSet. The endpoints of using this new set is clearly the most important one is DKA, prevention of DKA, and of course the regular safety endpoints like severe hypoglycemia, hyperglycemia, and adverse events and unanticipated device-related adverse events. The effectiveness endpoint was the survival. What's the percentage of catheters that reach 7-day use without failure as we have previously defined it? A definition of a set failure if we have an unexpected blood glucose of above 300 that is not corrected by more than 50 milligram deciliter after a correction bolus, or that this hyperglycemia is associated with some evidence of ketosis or a subcutaneous infusion set reaction. What were the results? First of all, let's look at the participants. The participants of this study were with a mean age of 45, but with an extended duration of diabetes of more than 21 years. They were equally distributed between the genders, and their A1C was pretty well controlled. The results of these almost 80 sets was that there was no safety issues, no DKAs, and none of the other safety measures that were followed. But let's look at the survival rates. What was interesting is that if you compare it to the QuickSet from the literature, and we can see here, this is a study again done by Bruce Buckingham's team in Stanford. When you use the QuickSet for 3 days, you do have a 3-day survival rate of around 77%. And after the 3 days, you have a rapid deterioration and accumulation of set failures. When you compare it to the survival of the patients in the study, interesting that after 7 days, the survival of the set was similar, a bit better than the survival of the QuickSet for 3 days. So the survival for 7 days was 80%. If you want to look at the amount of insulin use or utilization through the whole 7 days, we can see that there's a very stable use of the amount of insulin day by day throughout the whole 7 days. That points out that there's not a slightly decreasing site loss from the day 3 to day 7 that provides a very stable and a expectations that the pump and the infusion set will function fully for the whole 7 days. In this chart, we can see the gradual improvement and the progressive improvement in the sets from the current set to the new extended wear set. In the bottom line, the green line, is the survival sets from the historical studies done in Stanford, where you can see that there's a infusion set failure in the first day, and then the survival day at day 3 was around 77%. When we repeated these studies with EWIS, with the QuickSet, for 3 days and beyond, in a different site, we found a very similar line and a very similar survival of 3 days, but as this were adult population, there was less infusion set failure at insertion, but the cumulative failure after 3 days were actually plus minus 2%, close to the 77% at 3 days, and then the deterioration at day 3 and above. Using different iteration of the new set, you can see that in the blue line, just changing the cap increased the survival day of 7 days, and with changing the infusion set until we get to the last formulation of the quick set, we've reached a level of survival almost 80% at day 7, which is again better than the survival of the 3-day set at day 3. The conclusion is therefore that the extended wear system performs safely at day 7 just as well as the 3-day set is performing at day 3. And that is a proof that the extended wear actually meets the acceptance criteria for the intended use of these sets for 7 days. So to summarize what we have done, what we have discussed with you, we can see that Medtronic did provide innovative technology to improve the issues of stability of the insulin and results in less occlusion and consequently decreasing hyperglycemia due to issues with the infusion set. The infusion set actually survives twice as long as the current set and can be used for 7 days, and this can provide to save both insulin and materials. So I'm happy to share that after showing these results to the regulatory boards in Europe, this has been approved and has received the CE mark and is now going through a scale up for launch of these devices in Europe. In the US, there's currently a pivotal trial, which will be followed also by submission to the FDA, to the regulatory authorities in the US, and then subsequently launch. So with this, I would like to end and thank the EASD for the opportunity to provide you with this new novel technology and provide you the evidence for improvement of the infusion set for performances and patient satisfaction. And I want to thank Sarnath and you for attending this session. Thank you.